Great. Today we want to talk about sample preparation and imaging and things that we can do in the DualBeam FIB. All right. Let's see here. So today I want to keep the talk as brief as I can and save some time for questions and answers at the end. There's a lot of things that we can do in the FIB and a lot of capabilities that we have at CEMAS. And so, feel free to ask questions at the end. And also, you can contact me directly if you want to set up a time for a call or a chat over the web or whatever, if you want to talk about any applications you have for DualBeam FIB. At CEMAS, we have a number of different instruments, transmission electron microscopes, scanning electron microscopes. We're also very lucky to have 2 DualBeam FIB instruments. So about me, I grew up in Dayton. I've got a bachelor's and master's degree from material science, and then later on a PhD from an MSc as well. Worked for the CEOF, which is the microscopy facility before we moved to West Campus, and then now as an assistant director for CEMAS. I'm really excited about. I really am excited about making the best environment for transmission electron microscope, especially for high resolution imaging of atomic columns. So there's 2 things that really play into that: is the sample prep, but then also the environment. In our old facility, we're plagued with a lot of bad electromagnetic fields and whatnot. So characterizing those and determining kind of the environment we needed was a catalyzing force for moving to CEMAS, and that's where we're at now. So what is the DualBeam FIB and why do we want to FIB everything? It's pretty straightforward. There's focused ion beam instruments that have been around since the probably the 1970s for doing destructive testing and analysis on semiconductor materials. And then scanning electron microscopes have been around for a long time. And it was just natural to combine the 2, focused ion beam and scanning electron microscope into one instrument. That's our DualBeam FIB. So if we borrow from Steve's talk earlier on EDS, you get all the different signals that come out of the scanning electron microscopes. You got backscatter electrons, secondary electrons, X-rays, and photoluminescence systems. On the ion side, we have secondary ions and secondary electrons that come off, so we can generate an image just like we're going to be producing an image in a scanning electron microscope. You can do it with a focused ion beam. The main difference here, and so something that Hank and I kind of worked out one afternoon years and years ago, is if you're driving down the highway and an electron hit your windshield, and that was roughly the size of a bug hitting your windshield, how big would the gallium atom be? Gallium atom or ion when it hit your car? And we calculated to have roughly the mass of a locomotive. So that was the kind of the size difference, and why when you're blasting your sample with a gallium beam, you can actually remove material and not just take an image. So in our 2 FIBs, we have liquid metal ion sources. So the question is, why do we use gallium? Why is gallium a good source for the FIB? And the main attraction for it is this phenomena that's called a Taylor cone that's formed at the end of the needle. So it produces a very sharp, effective point source of ions. That sharp source gives us a very high brightness. It's like cutting with a sharp knife. That is a liquid interface phenomena. And so what's really interesting about gallium is it has a very low vapor pressure and a very low melting point. So it melts just a little bit above room temperature, but has a very low vapor pressure, so it's vacuum compatible. Gallium exists in 2 isotopes, 69 and 71. In our sources, they're actually a single isotope source. It has a low energy spread, gives us a very high spatial resolution. It's a relatively rare element. It's not found in very many minerals and some semiconductor and electronic materials. It's also a very heavy ion that produces an effective sputter yield, which means when the ion hits the source, or it's from the source hits the sample, it's going to blast away a lot of material fairly effectively. Second of all, the reservoir of gallium is behind the needle is supplying the source. It's got a significant reservoir of gallium there, so you can actually have a source that will give you up to 3,000 hours of lifetime. There are some other alternatives for ion sources. Gallium, like I said, is at low currents, you get a very good brightness. Gallium can react with your sample. You can embed gallium into your material. It can have some negative effects and have some changes that you need to be mindful of. So there's some other ions like xenon, which are very good. Xenon, the plasma sources are very good at high currents. At very high currents, they maintain a small effective source. So they give you high brightness at very high beam currents. However, at lower beam currents for making TEM foils and stuff, the gallium, the liquid metal ion source, gives you a little bit smaller effective ion source. So what are the state-of-the-art developments right now? And this is what we're kind of excited about at CEMAS is you know, some of the developments that have come out in this field. And so one of those is now Thermo Fisher is offering with a machine they call a Hydra, which is a plasma source system that is capable of running either xenon, nitrogen, oxygen, or argon ion species. Each one of these different ions has pros and cons and different abilities to mill with different materials. And we're excited to hopefully get one of these machines in the near future and be able to explore some of the possibilities of what we can do with it. So a question people always ask is like, well, how big is the FIB sample? Or, we often relate back to, like, well, how big is a human hair? If a human hair was so big, what would the sample be? And I've not been to the barbershop lately with everything being closed. I kind of thought this was like a good example to kind of work with today to give everybody kind of a feeling of, how big is a FIB sample and how it might relate back to their project. So here we actually have a human hair, and there's a little bump on the hair, and we're able to serial section into it. Turns out hair's actually got kind of an interesting and complex microstructure. There's actually a lot going on. So a collaborator of ours had reached out to us in a while back and said that they've been studying these little bumps on hair and said, would we be able to help characterize what the bumps are and what they're made out of. And so we really actually do it really successfully. Some of the initial things that they had looked at were just taking these hairs and slicing them with a razor blade to expose the internal structure. And you can see the cuticle layers and then also the cortex material and these little bubbles that were existing interstitial to the cuticle layers. And other attempts to take a look at this, they've done X-ray microtomography to look at the distribution of the bubbles on the outside of the hair. And they can see that they have a kind of a plate-like morphology, but we really couldn't see exactly what the internal structure was or anything. The other attempt that they had used was microtome, using a very sharp diamond knife, taking sequence slices from the hair to take a look at the internal structure. And what they found was that the microtome, with resin embedding and lifting out of the thin section through water, there was some degradation in the cortex material. And there was possibly also some little defects and things that were showing up inside of the cuticle layers. And that didn't seem to be, they were maintaining the highest fidelity in those layers. So we were able to take the DualBeam FIB, section into the outside of the hair, and be able to take a look on the inside and see that we're able to preserve the cortex and the cuticle material, and that we might be able to do TEM and other analysis using the FIB. So here we are looking at an area of interest. So we've selected a prominent little bubble on the surface of a hair. The hair has been attached to a carbon adhesive tab, and we gave it a quick gold coating to make it conductive for SEM imaging. We capped the area with platinum to protect it And now there's two different ways that we can go about starting our serial section. If you have a bulk material and you can go work on, say, your lab bench for a little bit, you can go polish the outside edges and prepare a nice sharp corner to start your serial section on. Or what we can do is we can kind of mill around the area of interest. If you look at the green specimen, where we'll just kind of trench into the material and expose an area that the electron beam can then image. So that's what we did in this case. We sectioned around the outside edge, exposing the area around the bubble that's been encapsulated in the platinum cap. And what we'll be able to do for sectioning is this is the sort of view where we would crop out all the rest of the information and just look at the area of interest for reconstruction. That reconstruction is going to look something like this. My video does not want to play completely manually, so what I can do is just kind of manually slide through. So you can see as the ion beam is slicing, we're taking an image each time, recording those images. We're going to use a program like MyPar and then Aviso to put all these stacks back together to kind of create a virtual 3D environment. Now it contains our sample. So we go back, you can see our little cortical layers, and there is that bubble internal. So now we're going to go back, and once we've captured that, we're going to go back and segment the area that has the bubble. We're going to go isolate everything that we think is the bubble. Now we're going to go build that up into a 3D structure. So now we got our 3D structure. We can now bring that back, rotate this model around, and for the first time be able to feel like you're going to pick up and take one of those little bubbles and really look at it and say, what is this thing? Why is it there? Why is this happening? What is it made of? You can actually see once we get to the bottom side, you can see kind of a little tab shape in the middle, and that's the interaction of that bubble with one of the lower cuticle layers, where it's actually that bubble is existing interstitial to the multiple different bubbles. The bubble is existing interstitial to multiple different cuticle layers. All right, so we know now with the FIB that we can see these bubble structures, and we kind of know what their morphology is, where they live, and what's going on. We still don't know exactly what they're made of. So the next step here is to go isolate one. We're going to go try and take a TEM foil from it. Now, because this is a soft material, we want to limit the ion exposure as much as possible. So I don't have very many pictures from the lift out of this particular bubble. But what I'm going to include here is a couple of example images from some specimens that were produced from regular materials or hard materials. And so first what we're going to do is we're going to trench on either side of that bubble. And we're going to cap the area with a little bit of platinum. And then you're going to back tilt the sample, use the ion beam to cut out sides and underneath of the specimen. And if you see on the picture on the right hand side, there's going to be a micro manipulator needle that we're going to use to pick up the lamella and transfer that onto a little Omni probe grid. That's going to be what we're going to use with our tweezers to pick up and transfer the specimen into the TEM holder. It's kind of the lower right hand corner. So once that sample has been put on the Omni probe grid, we can go back and we can do some thinning. And I think Steve and his EDS talk did a really good job showing here an example of a TEM foil where he has thinned it in a stepwise fashion, and then he scanned the sample with the electron beam at various different voltages and looked at the X-ray emission, one of the signals that comes out of the sample. And what you can see is as the sample is thicker, this is iron disulfide, you are getting some transmission of this through the sample at 30 kV, and as you go down in accelerating voltage from 30 down to 20, 10, or 15 and 10, the transmission keeps happening at the thinner and thinner parts of the specimen. So this actually highlights the trick that we use when we're thinning the specimen with the ion beam to check its thickness. So our trick here is we're going to view the sample over here on the left-hand side, in the SEM view at 5 kilovolts. And what's going to happen is, as the electron beam is hitting the specimen and we're thinning the ion beam simultaneously, as the sample gets thin enough that the beam starts to transmit through the specimen at 5 kilovolts, the contrast mechanism is forming the image, and the sample starts to change. And so the sample will start to appear bright when it gets really thin. We know that happens roughly around the mean free path of the 5 kilovolt electrons. It's going to be, in the neighborhood of about 100 nanometers in most materials. So as our sample is becoming a little bit less than 100 nanometers in thickness, we're going to see this brightness change with a 5 kV accelerating voltage. That's why we pick 5 kV. Steve's really kind of shown a good example here with his EDS data of where that transmission is happening, or where it would be happening there at 5 kilovolts. So now if we take a look at our bubble, and we're now in the transmission electron microscopes, this is going to be in one of the Titan instruments that Robert talked about last week with the Super EDS. So we're now going to map that whole specimen and look at all the x-rays that are coming out. And so in section A, we've got the high angular annular dark field detector, HADAF detector, forming our STEM image. And then in B, we're going to see a map of calcium K alpha emission. And then in C, you've got nitrogen, or C is nitrogen, D is carbon, oxygen, and sulfur. And so what we can see here is there's a considerable amount of calcium that's existing in this bubble, but not necessarily nitrogen, for sulfur is found on the outside of the surface, the outside surface of the sample. So what is that going to tell us? There's definitely calcium there. If we zoom in even further with the HADAF STEM imaging, we can start to look at the internal structure of each one of those little blob that's inside of that calcium-rich material. And what we see here in section C is there's kind of the same structure of a 7 to 20 nanometer lattice repeat that's happening. And so this is typical of what's been seen in other self-assembled liquid crystals. This has actually been seen in natural lipids. So this is giving us a good indication that this is a calcium fatty acid. These have not been studied very well because they tend to dissolve. It's hard to get a sample like this. So the researchers we're working with were very excited to be able to see the structure direct. So this was a really good example where we talk a lot of times about the gallium atom, the gallium ions coming down and doing damage into the specimen and what their interaction might be. And here we've got a case of very sensitive material. The lipids and proteins that are making up hair would be very sensitive to the beam exposure and whatnot. And here we're able to produce a very thin specimen and then also be able to preserve this liquid crystal mesostructure within the fatty acid. So this was really pretty exciting work on FIB quite a bit. And we mill inorganic materials and titanium alloys and nickel-based superalloys every day. And to be able to work on something that's this soft to beam sensitive and be able to maintain the structure is really pretty exciting. So to say, what else can we do with the FIB? I really liked this diagram that Thermo had put together for their sales information for their Hydra, the FIB. But it really shows a lot of the techniques that we have here at CEMAS. We've got microscopy capabilities, X-ray, CT, FIB. We're working on getting a plasma FIB. We're really big into the transmission electron microscopy. We can do atom probe sample prep. We don't have an atom probe, but we have collaborators that we work with that do. So we can prepare the samples at CEMAS and send them out for analysis. We can also do in situ TEM experiments. So we have some in situ holders. We also use the DualBeam FIB for these purposes. So the technique that I do is I prepare a sample like I normally do, put it on the OmniProbe grid, thin it, we can even check it in the TEM. And then what we'll do is transfer the sample from the OmniProbe grid back onto the in situ holder. So this is a sample that I produced for a collaborator in Chicago. And so this is a biasing, excuse me, biasing holder where there's a piezoelectric material in the foil, and the top and bottom of the foil are actually being biased at different potentials. And the thin area underneath the platinum cap where the piezo material is existing is now lined up over top of very small windows in between the two electrodes. And so the group that was doing this work was able to directly observe the piezoelectric effect in the material by applying a bias and actually seeing the atomic columns move. So it's pretty exciting to work on that. We can also perform a lot of experiments at cryogenic temperature. So we have a quorum cryo stage. Cryo stages are fairly common on scanning electron microscopes. They're less common on FIBs, but we're lucky to have one on our Helios. And the principle of doing the fibbing work is pretty much the same. The process is that we will take a sample, put that into liquid nitrogen, and then transfer that onto the stage, and then onto the cryo stage, which is being cooled by cryogenic gas, which allows us to work on the sample in a frozen and hydrated condition. I have another really reached out and said, you know, of the things that we're doing on FIB now that are pretty exciting here at CEMAS. Bob Wheeler has a microtesting solutions company, and he makes a very small micro manipulator which can allow users to do in situ mechanical testing on a very small size scale. And this has been done over the years in microcompression pillars. Paul Shade and Mike Uchek at the Air Force Base have been doing this at either at OSU or at AFRL for quite a long time. And the process is that we would take a very small piece of material and mill all the edges away until we come up with a microcompression pillar, and then in testing they actually push down on the pillar and measure the mechanical properties. And then over here on the right hand side, Bob would continue to make some micro actuators for actually pulling and measuring the strain on very small specimens. So he's got marked over here a 5 micron microcompression pillar, which is a little circle, and then he's got these micro tensile specimens he's produced in 1A and B. So actually kind of like a little gripper they can pull a specimen. So here's an example of a microcompression pillar that's been tested and uniaxially loaded. This is Paul's work back in 2005. And then over here, this is a micro tensile specimen. The grips have actually pulled the material. You can see the formation of the shear bands and whatnot. So really interesting if you want to go study these different arcs. Now you've got the strain, you know, the loading, it's all confined. You can actually go back and create a TEM foil directly from the specimen and get a look inside. Here's an example of, let's see if this will play here. This is a copper material that gets pulled and stressed and stretched all the way to failure here. You actually start to see all the grains start to come apart. The material will neck down and then ultimately break. And so this is, again, all happening on a size scale of only a few, tens of microns here inside of the scope. So this has been really useful for studying interfaces and weld materials. Dissimilar joining and stuff where you've got two different materials. It'd be almost impossible to go back and test in bulk what the material at the interface might be in terms of its strength and properties, where now with the FIB we can go select a very small area, say of a weld, and then test that material and find out what its properties are. That's useful for putting that information back into models for predicting what the properties and stuff might be. And so those are some of the things that we can do.